Good afternoon. Thank you for the opportunity to be back here again. Wonderful to be back in person, I think, for all of us. I'm happy to give you an update for those of you who are familiar with our story and to introduce Sentien to you for those of you who might not know of us. We are an early-stage clinical company, cell therapy-based. We're developing allogeneic mesenchymal stromal cells, or MSCs, for treatment of systemic inflammatory disorders and indications characterized by systemic inflammation. Our twist on MSCs is a little bit unique. We've been focused on trying to solve some of the bioavailability challenges that have hindered somewhat of the clinical applications. And the approach that we've taken is really to make a combination cellular device where the MSCs actually never enter the body. So instead of injecting them or infusing them, we're actually bringing the blood to the MSCs, enabling the blood and the MSC secretome to interact with each other over a much more controlled, sustained period of time, really optimizing the bioavailability and the pharmacology profile. I'll get into more of that in a little bit later. As I mentioned, we're a clinical stage company. I'm happy to share a little bit of early clinical data with you that we think is really supportive of the overall therapeutic hypothesis. We're happy to also announce for the first time at this conference partnerships with BARDA and with Maryland Stem Cell Research Foundation to support some of the clinical activities we have going on. We are also looking for corporate partnerships to really accelerate and expand a lot of the opportunities that we have, as I'll describe in a little bit. So one of the things that COVID did is really brought the idea of systemic inflammation, cytokine storm, into the forefront. I knew when my mother asked me about cytokine storm, it had crossed some certain thresholds. But that's really what we've been trying to treat for a long time. It's really this dysregulated host immune response causing hyperinflammation that underlies a lot of different indications, specifically in the critical care area. Very difficult to treat, very complex biology, complex pathophysiology, and with that, we think it's a real opportunity for a cell therapy to shine as a complex medicine. So MSCs have been long known to be potent immunomodulators, and so what we set out to do, again prior to COVID, was think about how we could deliver the MSC secretome, which is really the underlying mechanism of action for reprogramming the immune system away from this dysregulated response into more of a wound healing phenotype. So the engineering behind this is really trying to allow for sustained, controlled exposure of the MSCs to the immune system via the blood, while also enabling the cells to respond dynamically to their microenvironment. So people are using, in really cool ways, EVs and those types of microvesicles as a way to deliver a static medicine. In our approach, we're still enabling MSCs to sense their microenvironment and in a time-specific manner think about how they can change their secretome. And I'll show you some data that we've just recently published that shows we're actually doing that. One of the cool features of our approach, because the cells never enter the body, I'll describe the clinical application, we can actually sample from our product during treatment. So we can watch, obviously, how the patient's doing, but interestingly, we can also observe what the MSCs are doing while they're doing it. So that creates a new data set that really has not been explored, and I'll describe that in a couple of slides. The clinical application is incorporating this into clinically utilized blood circuits already as part of standard of care. So our first indication that I'll describe is acute kidney injury. The standard of care today for severe AKI patients is called continuous renal replacement therapy. So there's already catheter-based blood access. We then basically just create an add-on to that existing standard of care. So SBI 101, our product, can be integrated with an ongoing dialysis circuit, thereby providing the underlying dialytic therapy along with our active treatment addressing this dysregulated immune response. So it's really that sort of critical care setting that we focused our initial efforts. Where we are, we completed a low-dose cohort in AKI requiring dialysis prior to the pandemic. Once the pandemic hit, we then focused over into a COVID-induced AKI. We're really agnostic about the etiology of the systemic inflammation. Completed that low-dose cohort after a Herculean effort by the team to get the new IND opened. And now we are starting enrollment in a slightly broader indication. It's almost a merger of the two previous studies where now we're looking at sepsis-induced AKI. So there's a lot of regulatory work that I'm very proud of that I won't get into today. But moving forward, now we have a regulatory path forward looking at sepsis-induced AKI, regardless of the type of infection, enabling then, again, a sort of a during COVID, but also post-COVID opportunity to continue forward. So we're starting with our high-dose cohort, and we're excited to see what that data might look like. So what I'll show you on the next 4 slides is some of the early preclinical data, very small numbers to be sure, but even with that, we were very, very encouraged by the magnitude of effect we were seeing even at our low dose. As I mentioned, we can sample from our product during treatment. What I'm going to show you here is evidence supporting both the duration, the viability of these cells throughout the duration of treatment, and also the dynamic responsiveness. So on the top, and I know you can't read it, but I'll point you to the publication that has all of this and more, shows that over the entire 24-hour duration of exposure, which is our initial dose, that these patients are receiving into the bloodstream MSC-secreted factors. And so that's really what we're showing on the top line, that they're viable over the entire duration of treatment. What's interesting on the lower panel is, again, this dynamic responsiveness. So on the top, what you see is different patient profiles coming into treatment. And on the left-hand side is active, on the right-hand side is sham. There were 4 in each group. On the left-hand side on the bottom in the blue is a heat map representing different MSC-secreted factors that we measure from the device. So what you can see, again, is that each patient has a unique inflammatory profile, and the MSCs are dynamically responding during treatment with a tailored secretome in response to the microenvironment. So the MSCs are sensing and then dynamically responding to what the patient is represented in their blood. So all that suggests a lot of heterogeneity on the front end. What we were pleasantly surprised and hopeful to see is more of a consistent response on the back end. So even with all that heterogeneity going in, we saw a pretty conserved response biologically on the pharmacodynamic side. And that response also was consistent with very well-characterized MSC biology. So again, without going into all this, it's in the paper, there's clear distinctions of a reduction in pro-inflammatory cytokines, representative like Th1 profiles, and an upregulation in Th2-related cytokines. We saw a clear response on the innate immune system as represented by different populations of monocytes. And importantly for the actual tissue, saw a significant, not statistically significant, but a notable reduction in kidney injury markers. So in the kidney, if you have ongoing apoptosis and necrosis, there are markers that go out. And what we saw is a clear reduction of some of those injury markers in response to our treatment. Again, all this was at our low dose, very encouraging, again, to see that type of consistent response with what MSCs are known to do. Stepping back from sort of single biomarker types of profiles, we looked at over 200 cytokines and really started asking questions about broad immunomodulation. So what's shown here, again, on the left-hand 4 columns are the 4 active subjects. On the right-hand side are the 4 sham. What we're doing here is a pathway analysis. So over 200 cytokines being evaluated. Does that suggest collectively an upregulation or a continued activation of pro-inflammatory pathways, or conversely, an inhibition of some of these pro-inflammatory pathways? And what we saw in the active was a clear, consistent, and I say consistent in that it happened in every case, inhibition of a lot of these pro-inflammatory pathways. Again, what you would hope to see in a complex biological setting and pathophysiological setting like the indications that we're treating. On the right-hand side, where these sham patients were not getting anything but a sham device, 3 of the 4 showed a continued activation of a lot of those pro-inflammatory pathways. The one exception was almost something that we could not have planned for. It was a subject randomized into the sham group, but was in the resolving phase of sepsis during activation or during the treatment with sham. So she was naturally resolving her inflammatory profile. So what that provided with us is what a natural wound healing response looks like from pathway perspective. And we were very pleased to see that it mirrored what SBI 101 was doing in a much more consistent basis. So it gave us almost like a roadmap of what healing looks like at the sort of pathways level from the immune system. Bringing it back to the tissue bed, it's important to understand how this would impact the organ itself. Again, looking at markers of early and late injury relative to markers of wound healing and regeneration, what we saw in active versus sham was a clear downregulation of these injury markers and a clear upregulation of wound healing and regenerative markers, suggesting if you sort of step back and look at it, we're accelerating these patients through the injury phase into the wound healing and regeneration phase. So all this paints an early pharmacological profile that the engineering is working. The cells are doing what we would expect them to do. Patient numbers are all obviously very low. So the question becomes, what do we have in terms of clinical outcomes? So going into the COVID study, again, I can show you 4 patients on active here, each treated for 10 hours or more. And without going through all of it, what I'd highlight is COVID patients that are on both a ventilator and requiring continuous dialysis have a mortality rate expected to be greater than 60% at day 28. With our treatment, small numbers, but N of 4 all survived out to day 28. And in fact, most of them survived quite a bit longer, and a few were even discharged to home in a relatively quick manner. So the clinical outcome story is encouraging. Obviously, we have to have much larger sample sizes. We're excited to see what the high dose will do in terms of both the biological profile and hopefully continuing on the clinical outcome side. And as I mentioned, we are enrolling right now in the high dose cohort for SBI 101 in sepsis-induced AKI. We're pleased again to partner with both BARDA in their Drive Solving Sepsis team. They define sepsis as sort of a systemic infection, systemic inflammatory response driven by a dysregulated host response, very much in line with how we think about it, and more recently with MSCRF also to support the ongoing effort. The goal with this program is really to get a clinical candidate for a phase 2 proof of concept study in a systemic inflammatory indication in the critical care setting. With that said, we have much broader ambitions and visions. On the pipeline side, obviously, we're focused on the lead program and demonstrating this pharmacological proof of principle at the high dose to complement the low dose data I've showed you today. We think there are other applications in a tissue agnostic manner, so liver, lung, whether it's blood for basic sepsis. I say basic sepsis as if there is such a thing. But all of these critical care indications have this common systemic inflammatory profile that really drives a lot of the morbidity and mortality. And that's really where we think we can make near-term impact in a number of different indications. We do have early preclinical data on the chronic side. We'd be excited to share that with you if of interest in a less time-constrained manner, where we can think about modulating autoimmune disease, neuroinflammatory disorders, again in a similar type of manner, where the modality then could be contemplated even in the context of like an outpatient dialysis center for delivery for very severe types of neurodegenerative diseases with inflammation at the core. So we'd love to talk to anybody who's interested in these spaces. As I mentioned, we are looking at corporate partnerships to both accelerate and expand the opportunities. Right now, we're pretty focused on the lead program and getting through that, generating that data, but we're very ambitious and excited to move on, continue that work, but also expand and accelerate some of the other opportunities, particularly in the chronic side where we think there's untapped potential. So the clinical data that we've shown, we believe, is really encouraging with small sample sizes. It's exactly what we wanted to see. We're excited to see what the high dose can offer us as well and how that might differentiate and expand on the low dose data. And if anybody would like to talk more, again, I encourage you to read the paper for the fuller and more articulated story around the data than what I presented here today. And I'd love to chat with anybody afterwards. So thank you for your time.